Hello children, welcome back to biology class. I'm very happy to meet you all after the long break. Today we are going to learn about reproduction in plants. What is reproduction and why it is necessary? It is a vital process for existence of species. It brings changes through variations in the offsprings for the survival. It plays an important role in evolution and reproduction categorized into two types. They are sexual and asexual reproduction. Let us learn about the types of asexual reproduction in plants with examples. First one is conidia. Conidia formation is seen in the plants called aspergillus. Next, budding. Budding is seen in yeast. Next one is fragmentation. It is seen in Spirogyra. The next one is production of gemmae or gemma. You can find this type of reproduction in the plants called Machantia. Next, binary fission. Bacteria shows the binary fission reproduction. Next, regeneration. Planaria shows regeneration. Now, these are some of the types of asexual reproduction found in the plants. Clones are also produced during asexual reproduction. Here, the individuals are formed by this artificial method and it is morphologically and genetically identical. In the higher plants, asexual reproduction is done by vegetative reproduction. There are two methods of vegetative reproduction. One is a natural method and another one is called artificial method. These are the two types of asexual methods reproduction in the plants. Let us learn about the natural method. Now, the natural method is a form of asexual reproduction. Here, the bud grows and develops into new plants. The buds may be formed in roots, stem, or leaf. The structure is called reproductive propagules or diaspores. Let's watch the video and learn more about natural method of reproduction in plants.
Now let us see few examples of vegetative propagules in angiosperms. A. Eyes of potato. B. Rhizome of ginger. C. Bulbils of agave. D. Leaf buds of bryophyllum. E. Offset of water hyacinth. Rhizome. Modified stem grows horizontally. Distinct nodes and internodes are present with marked apical and axillary buds. Adventitious roots present at the ventral side. Example: ginger, gingiber, turmeric, and curcuma. Tuber. In Solanum tuberosum, half-moon-like structures called eyes of potato. These eyes indicate the position of nodes and internodes. Scale leaves and axillary buds are present. Apical bud is also present. Tubers have no roots. Example: Solanum tuberosum, potato. Heliander tuberosus. Onion. Underground stem modification. Adventitious roots grow towards the soil. Fleshy, scaly leaves, which are eaten, grow above soil, aerial. Auxiliary buds produce new plant. Apical bud shows inflorescence. Tunicate bulbs are seen in Allium cepa, onion. The imbricate bulbs are seen in Allium sativum, garlic. Corm. Underground stem modification grows vertically in the soil. Nodes and internodes present. Scale leaves are rising at node. Apical and auxiliary buds are present. Auxiliary roots produce daughter roots. Comb shows contractile roots, which are helpful to put the comb in distinct depth. Example, Amorphophallus, Colocasia, etc.
Let's learn the advantages and disadvantages of natural method of asexual reproduction. The advantages are the only one parent is required. The new individuals are genetically identical. Some plants grow and spread very rapidly. It is utilized by farmers and horticulturalists to harvest the crops in large scale. Let us look at the disadvantage. The new plants have no genetical variations. Let's learn of the second type, the artificial method of asexual reproduction in plants. This is used in agriculture and horticulture to propagate plants from their plant parts. The two methods of artificial methods are, number one is conventional method. And the second method is called modern method. Conventional method and the modern method. Conventional method is a method have been practicing for a longer time. The three types of conventional methods are cutting, grafting, and layering. Let us watch this video and learn about method of cutting.
Small vegetative part of a plant removed by making a cut with gardening scissors is called a cutting. A cutting may be a piece of stem, root, or even a leaf. While making a cut, care should be taken to see that there are some buds on it. Stem cutting is commonly used for propagation of grapes, rose, sugarcane, etc. In this method, a cutting of the parent plant, stem or shoot, having some buds on it is taken, and its lower part is buried in the moist soil. After a few days, the cutting develops roots and grows into a new plant. Buds of potato and leaves of bryophyllum are also used as cutting. The new plant formed by cutting is exactly similar to the parent plant.
Hope you understood about the cutting. Let us learn about the grafting, the second type. Here the two different parts are joined. The stalk is a part contact with the soil, that is the lower part. The upper part is called scion. The scion is a part used for grafting. The types of grafting are bud grafting, apical grafting, tongue grafting, crown grafting, and wedge grafting. Example, citrus, mango, apple, etc. Let us learn one by one in detail. The first type. In bud grafting, the T-shaped scion is made in stalk. The scion as a little wood act as a bud. Both the scion and the stalk are bandaged with the tape to form the bud grafting. Next, we'll go to the next type called approach grafting. Here, the scion and stalk with same thickness remain rooted. The small size is cut and brought closer and they are tied with a bandage. After 1 to 4 weeks, the tip of the scion or the plant detached from the parent plant and grown separately in the soil. The next type is nothing but tongue grafting. In the tongue grafting, the scion and stalk of same size are cut obliquely and the scion is fit into stalk and tied. That's how the tongue grafting is made. The fourth type is crown grafting. Here the stalk is very large size. The scion is cut into wedge shape and inserted using graft wax. The last type is nothing but wedge grafting. Here the slit is made in stalk. A twig of scion is inserted so that the cambium is joined to form wedge grafting. These are some of the methods of grafting. Let's learn the third type of conventional method called layering. In layering, the stem of the parent plant is allowed to develop root which is still intact. Rooted plants are cut and planted to grow into new plant. Example, jasminum. There are two types of layering. They are mound layering and air layering. Let us look at the mound layering. In mound layering, the applicable for plants with flexible branches, the lower branch is bent to the ground, buried into the soil, and the tip is exposed above. After the root is emerged from the rooted plant, it is cut and grow into a new plant. In the mound layering type. The next type is called air layering. In the air layering, the stem is girdled at nodal region and the hormones are applied to grow the root system. This portion is covered with damp soil using polythene sheet. The roots emerges in these branches after 2 to 4 months. Finally, the branches are removed from the parent plant and grow separately into a new plants. That's what's happening in the air layering. Let us look at the advantages and disadvantages of this conventional method. The advantages are the plants produced are generally uniform. It can be produced very quickly in a very short span. With the help of conventional method, disease resistant and high yield plants are produced. The disadvantages are virus infected parent plant produce only the virus infected new plants. The vegetative structures are bulky, difficult to handle and store for a long time. These are some of the disadvantages and advantages of conventional method. Let us go to the next method called modern method. The modern method was discovered by the Professor C Steward in the year 1932. There are two methods, tissue culture and micropropagation. In tissue culture, the growth of plant tissue in special culture medium under suitable control conditions are produced. The plants are grown under the suitable conditions. In the micropropagation, the regeneration of whole plant from a single cell, tissue or a small piece of vegetative structure through the tissue culture. These are the two types of modern methods used during the plant reproduction. Let us learn about the disadvantages and advantages of modern method. Now, what are the advantages? Here, the plants with desired characters produce in short duration. The plants are genetically identical. The tissue culture can be carried in any season, whether it's summer, winter or rainy season. Next, the endangered species and the disease-free species are produced or propagated. Some of the disadvantages are it is labor intensive and requires skilled worker. It needs many number of laborers and skilled workers also. Expensive due to the maintenance of sterile condition, because we need to maintain the sterile condition for a longer time. Finally, the callus may undergo undesirable changes. We can't predict what kind of a change happens to the callus. Sometimes it might undergo undesirable changes. These are some of the advantages and disadvantages of the modern method. Let's recall what we learned in today's class. First, we started the topic reproduction in plants. Later, we learned about the asexual reproduction, the methods and the examples. Then, vegetative reproduction, the natural and the artificial methods of vegetative reproduction, and a natural method by a root, stem and leaf with advantages and disadvantages. Under artificial, we learned about conventional method and the modern method. Under conventional method, we discussed about cutting, grafting and layering. Under modern method, we have discussed about tissue culture and micropropagation along with advantages and disadvantages. That's all for today. Goodbye.